welcome back, guys. I'm here for the second part of this structural systems biology talk. And so what I'm going to be talking about today are mainly the structural proteome. So this is how we can compute with protein structures on a genome scale and the pipelines we've developed to do this. And we're going to dive into some recent applications of the last 10 years or so on GEM-PROS, which are what we call these genome scale models with protein structures. So like I just said, that's what we've named them. So that comes from GEM-PRO here. And the first application came about in 2009 when a structural genomics project for Thermatoga maritima was completed. So as I mentioned in the last lecture, the aims of a structural genomics project are to solve all the protein structures within a genome. And so they had done this for Thermatoga maritima, which is a hypothermophilic organism. And they had all these structures, but they wanted to give them a context. And the context, luckily, was a recently reconstructed metabolic network. And so what they did was they took the enzymes that they had solved structurally, they assigned them to the reactions that they carried out within the metabolic network, and they wanted to understand a pretty basic evolutionary question, which was how are protein folds distributed throughout the metabolic network. So I'll talk about this in the application slide, but that was kind of the first application or the first generation of a GEM-PRO, which are the application of protein structures within reconstructed metabolic networks. And so again, the concept here is simple, that we are representing the enzymes with the 3D structures of these proteins. And so the main goals of a pipeline that we've recently created, so we're calling this the GEM-PRO pipeline, is to apply the same rigorous reconstruction process that we have to reconstruct metabolic networks to the structural proteome. So as you can imagine, there are either a lot of protein structures solved for your enzyme or interest, or there may be none at all. And you're going to have to do a kind of gap-filling process to predict these structures that do not exist yet experimentally with homology modeling techniques. Or you're going to have to figure out what enzyme structurally best represents a reaction within my metabolic network. So the GEM-PRO pipeline aims to answer these questions with a rigorous process to do that reconstruction. And the other half is to allow systems biologists to compute with protein structures at a genome scale. And what I have here is just a figure of kind of the applications, the different scales of applications you can do with the GEM-PRO from the bottom up to the top down. So I consider the bottom up kind of more using structures one by one to understand, say, what an enzyme's true function is within a metabolic network. Whereas a top-down approach would kind of consider all your gathered proteins in, say, comparative analyses to understand why this proteome differs from another. And so on the right here are some software links that are accessible to you guys in terms of running the GEM-PRO pipeline or looking at specific application notebooks, Jupyter IPython notebooks that we have, as well as pre-made GEM-PROs for human, which recently came out, and E. coli, and the newest reconstruction of E. coli. So just real briefly, the goals, again, of the GEM-PRO are to get a high-quality structure for an enzyme within your metabolic network. So how do we go about doing that? And so early on, it's really a simple ID mapping process where you try to either blast the protein data bank for sequences and structures that match what you're looking for. And if you don't have anything, you go through homology modeling processes to generate structural models for your enzymes. And one of the main techniques that we've been using is ITASR, but there are a number of other homology modeling pipelines out there that can be used. So once you have that, you either have one or many structures to represent an enzyme within your metabolic network. And what you can do then is check the quality and run it through some QC/QA checks and understand if the sequence of that experimentally solved structure matches your organism of interest, or if it's from another organism, or something like that. You can check its quality in terms of the resolution, which we mentioned in the last lecture, or a number of other quality metrics. And so once you do that, you can either rank order your list of multiple PDBs, or you can just pick a final homology model to use. And finally, we have this kind of refinement workflow where we take, if a structure doesn't fully match what our sequence is in our organism, we have a refinement workflow to take that, run it through some really short MD type simulations to correct it to the wild type sequence and get our final structure. So this was kind of developed in the last three years or so. And this is now available to the public as the GEM-PRO pipeline. And so what we did first was we used this to redo the Thermatoga study as well as a previous E. coli GEM-PRO. And here is just a pie chart of the coverage of metabolic enzymes, metabolic proteins, with experimental structures in green and homology models in blue. So today is actually up to 50% coverage in terms of structural coverage for the metabolic network, which is pretty good. So you can see that every couple of years or so, you're increasing about 5 to 10%. And hopefully one day we'll have a lot more of this covered with recent advances in membrane protein crystallography and cryo-EM. So that's one half of the challenge, is building the GEM-PRO and getting it to a high quality. The other half is making it available for systems biologists to use, and reproducing these results and disseminating it. So those were the goals of the SS Bio Python package that I recently released. And here, what you can do is you can easily compute all of these properties that we talked about last lecture, like solvent accessibility, secondary structure, thermostability. You can do that per protein in the context of a metabolic network. So what you can do is you can either simulate your network based on these properties, or even maybe simulate them with these properties while you're doing the simulation. So that's the end goal here. So now it's easily possible to do all these ID mappings, predict these properties either from sequence or compute them from structure and apply them directly in genome-scale models. And so with that, I hope it makes it easier for people to do structural systems biology on a large scale. So there are a couple of things I didn't mention as part of the GEM-PRO pipeline that we are currently working on and we would like to consider for further expansion. And one of them is protein complexes. So this is the quaternary structure that we were talking about in the last lecture, which is the active form of a lot of enzymes in a metabolic network. So here is just the construction of ATP synthase coming together to form its active complex, and each of the repeated frames here are single protein tertiary structures coming together, and if they're a different color, they're actually a different protein coming together to form the active ATP synthase. So you can see that there's a ton of different components that need to come together for it to finally form its active complex. So this is kind of a difficult challenge to tackle, just because there are so many possibilities for a stable form of a complex. There are inconsistent annotations for things like this in databases. And what we can do is we can, at least in M models, these do not need to be considered as rigorously as we need to do in me models, which I think you guys will learn about in the next lecture or so. So me models do consider things like complex stoichiometries and the assembly reactions that are needed to create a protein complex in your model. And so protein structures will come into handy very well there. So there's a good resource on the PDB, so it's called the PDB 101, if you want to learn more about these types of things. They have some really good articles on what protein complexes are and how they're represented in PDB entries, because they can be different. A PDB entry can have a single subunit of a complex, which may have to come together to form a biological assembly, and that may have to be computed before you can actually assemble the final complex from the original subunit. So these are things to consider when you're putting complexes together structurally. Another interesting thing that we're working on are membrane proteins within the cell. And so, at least in E. coli, membrane proteins are pretty interesting economically, because there is a trade-off between the cell and growth when it's at a very large size. So there's a trade-off when you're growing very fast catalytically, and what they found was you switch, an E. coli cell will actually switch to ferment instead of respirate, which is interesting because you think that at a higher catalytic rate, you'll want to produce more ATP through respiration, not fermentation. But the interesting question here is that this may be related to the surface area of the outside of the cell, to its volume. So at a higher catalytic rate, you're going to have a much higher volume than surface area that's available, where it may be more efficient to use fermentation to produce your ATP because respiratory enzymes are taking up a lot of space on your surface area. So these are kind of the questions that you can start to answer with structural information, because what structural information will give you is the information about the space that a membrane protein takes up on the surface of a cell. So reconstructing the membrane becomes a priority with this kind of question to answer. So finally, what we have with a GEM-PRO is the ability to instead compute properties in terms of a matrix with residues as the rows and different properties as the columns. We can do the same thing except for a whole proteome with proteins as the rows and the properties of these specific proteins as the columns. But we can also consider these when we're computing with proteins locally. So we can take actually both of these kinds of matrices, compute with them at the genome scale, and use them for different applications, which I'll talk about next.